Biological drugs have proven to be extremely specific and effective, but because they are applied systemically, they can cause severe side effects. This year, Slovenian iGem team decided to overcome this problem by engineering a novel delivery system for biological drugs. To present the problem of conventional delivery, we made a case study of hepatitis C, which is an infectious disease caused by the virus which primarily infects the liver. It is a serious worldwide health problem as 170 million individuals are infected. Currently used therapy relies on interferon alpha, a protein which helps to combat against the virus. It is injected 3 times a week, which presents a burden for the patient. After subcutaneous injection, it goes into all of the body organs, as you can see on this animation. The distribution of the drug throughout the body accounts for its side effects.
Our idea was to engineer therapeutic protein producing cells and implant them directly into the affected tissue of the patient. We should microencapsulate these cells to protect them and to avoid an immune response. Then we could use external signal to trigger the production of a desired therapeutic protein, and not only that, we should be able to switch between the production of one to another drug by a second external stimulus.
With this system incorporated in therapeutic cells, we can achieve advanced therapies with different drug combinations. In our case of hepatitis C, we can first produce interferon alpha, which helps eradicate the virus, and then switch to produce another drug. Our candidate would be hepatocyte growth factor, which helps regenerate the liver damaged by viral infection.
To achieve this, we needed to design multiple genetic switches inside a single cell. We choose TAL effectors because their DNA binding domain can be engineered against almost any nucleotide sequence. Even more importantly, their biochemical properties are very similar, which makes them nearly ideal building element for our switches. By coupling TAL DNA binding domain with mammalian effector domains, we were able to design such a genetic switch. Because TAL effectors bind to DNA in a monomeric fashion, we needed to introduce positive feedback loops to make sure our system exhibits non-linear response. Now let's see how it works. When we add an inducer molecule, it binds to its corresponding repressor and they dissociate away from the DNA. This triggers the transcription of TAL activator that activates one state of the switch and repressor that represses the other. Inside an activated state, additional pair of TAL regulators is transcribed, causing the positive feedback loop and the additional repression of an already repressed construct. This way, even after the removal of the inducer, the system remains stable.
We used our pharmacokinetic model to compare our novel therapy with current treatment. We can see that with our system, interferon alpha stays confined to the liver to a greater extent. Moreover, its concentration does not rise and fall in regular intervals, but stays constant, which is better as concentrations higher than needed also contribute to adverse effects. Although we presented how this novel delivery system would work in a hepatitis C patient, we really designed our system to be universal. This means it is possible to engineer cells to produce virtually any desired biological drug. We believe there are multiple diseases where our system would prove beneficial, such as ischemic heart disease, peripheral vascular disease, and complicated bone fractures, or wounds that don't heal properly. Besides offering a brand new approach to treatment of various diseases, our newly designed genetic switch framework has an even broader range of applications. We could put multiple orthogonal switches inside one cell. In that case, one switch forms a basic building block of a scalable epigenetic memory. It could be also used to run various complex cell programs, transforming our cells into next-generation biological computers.